At sunset, a living layer rises through the ocean, turning darkness into a moving continent of animals. Contains fishes, crustaceans, gelatinous predators, and countless zooplankton ascending from daytime refuge. Before dawn, much of that layer descends again, completing a journey repeated across enormous ocean regions. This is Diel vertical migration, often described as Earth's largest animal migration by total biomass. If you floated above it, the surface might look unchanged while billions of decisions unfolded below. The mystery is why such different animals synchronize vertical travel and what their movement changes. Light provides the clock, predation supplies pressure, and food near the surface offers a nightly reward. Each migrant must trade a productive feeding zone against exposure to hunters guided by vision. Our investigation follows one cycle from fading daylight through ascent, feeding, descent, and daytime concealment. The moving animals do not form one coordinated army, and their routes vary by species and location. Some travel hundreds of meters, others shift only within the upper ocean, and some migrate in reverse. Yet, conventional night ascent is widespread enough to create layers visible to acoustic instruments. Early sonar operators mistook these dense scattering layers for false seabeds that moved with twilight. The apparent bottom was biological, composed of gas-filled organs, bodies, and structures reflecting sound. As the sun lowers, irradiance declines through surface waters, and the risk from visual predators changes. Small plankton detect the dimming cue, begin swimming upward, and enter layers richer in edible phytoplankton. Their ascent increases feeding opportunities, while darkness reduces detection by many daytime fishes. Lanternfishes and other micronecton follow prey upward, carrying the migration into higher trophic levels. A lanternfish reads changing light, rises with the scattering layer, captures zooplankton, and stores energy. Before sunrise, brightening water raises exposure, prompting descent toward dimmer daytime depths. This cue-action adaptation sequence is supported by repeated acoustic records and net sampling. It is not universal because hunger, clouds, moonlight, temperature, and oxygen can shift timing and distance. Many migrants appear to follow a preferred light environment, rather than one fixed clock time. As light penetrates differently through clear and turbid water, the migrating layer adjusts its vertical position. Moonlight can delay ascent, deepen nighttime residence, or reduce surface activity in some communities. You would see only a faintly illuminated surface, but small eyes may register decisive differences. Laboratory experiments reveal responses to light, while field instruments show whether those responses scale naturally. Neither alone proves the same rule for every animal inside a broad acoustic layer. Temperature creates another boundary because surface warmth raises metabolic demand and changes muscle performance. An animal entering warm water may feed faster but spend stored energy more rapidly. Descending into cold water slows many physiological processes, potentially conserving energy during daylight inactivity. This thermal benefit complements predator avoidance, although its importance varies with body size and species. Oxygen minimum zones can compress migration routes or provide refuge from predators unable to tolerate low oxygen. Some migrants possess enhanced oxygen transport or metabolic suppression that permits temporary entry into these layers. A squid descends into depleted water, reduces activity, avoids certain hunters, then returns upward to feed. The refuge protects it, but oxygen debt and limited performance impose physiological costs. Where low oxygen zones expand, migrants may crowd into narrower bands and alter predator-prey encounters. The migration scale first becomes visible through acoustics, which records echoes across time and depth. Shipboard echo sounders send sound pulses downward and measure reflections returning from biological targets. Different body structures scatter frequencies differently, and abundance estimates require careful calibration. Net samples provide physical identification, but can miss fast swimmers, fragile animals, or precise depth boundaries. Optical systems record form and behavior, although lights may attract, repel, or overwhelm some animals. Using these three methods together strengthens interpretation while preserving their separate limitations. A bright echo could represent many small organisms, fewer strong scatterers, or a changing mixture. Researchers therefore avoid turning acoustic intensity directly into biomass without local biological information. Global estimates extrapolate from limited tracks and models, leaving uncertainty around the title's superlative. What is verified is the migration's broad distribution, immense abundance, and consistent daily rhythm. Why it evolved is tested through competing predictions about food, predators, metabolism, and ultraviolet exposure. Predator avoidance predicts deeper daytime residence where visual hunters become less effective. Feeding opportunity predicts ascent toward productive surface layers when darkness reduces the associated risk. Observations frequently match both predictions, making them complementary rather than mutually exclusive explanations. Metabolic optimization predicts time split between warm feeding water and colder resting depths. Experiments and models support this for some animals, while others remain active throughout the cycle. Ultraviolet avoidance may matter near the surface for sensitive organisms, especially during daylight. No single hypothesis explains reverse migrants that rise by day and descend at night. Reverse migration can reduce encounters with predators that themselves perform conventional night ascent. A small grazer tracks safer daylight conditions after its principal hunters leave, then retreats when they return. The cue is predator presence. The action reverses the common pattern, and survival may improve. Such behavior proves the migration is a food web strategy, not merely a reflex to sunset. Hunger can override caution when depleted animals ascend earlier or remain feeding longer. Well-fed individuals may stay deep, producing partial migration within the same population. Age and body size also change swimming capacity, diet, and vulnerability to specific predators. You could sample one depth and mistakenly label an entire species absent while another life stage passes elsewhere. The nightly movement transforms more than the location of animals, because feeding and respiration occur at different depths. Migrants eat carbon-rich material near the surface and metabolize part of it after descending. Respiration, excretion, and mortality then release carbon below the layer where it was consumed. This active biological transport complements passive sinking by moving matter inside living bodies. Some carbon returns quickly through circulation, while some remains isolated from the atmosphere for longer periods. The amount depends on migration depth, metabolism, mortality, and the physical movement of water. Global carbon estimates carry substantial uncertainty because migrant biomass and physiology are unevenly measured. Even so, Diel migration is recognized as an important component of ocean carbon cycling. It also moves nitrogen and other nutrients, linking food web processes across vertically separated habitats. Predators time their own movement around the layer, creating a daily pulse of encounters. Dolphins, tunas, seabirds, and larger fishes may exploit migrants where geography brings them within reach. Commercially important species can depend on mesopelagic prey, even when humans rarely see that prey directly. Fishing the deep scattering layer could therefore remove forage and alter active carbon transport simultaneously. Proposed exploitation of mesopelagic fishes remains constrained by ecological uncertainty and technical difficulty. Artificial light at night affects coastal plankton movements, though open ocean consequences are less well established. Climate change may modify stratification, productivity, temperature, and oxygen boundaries that shape migration routes. When oxygen habitat contracts, predators and prey may overlap more intensely within compressed layers. When surface food shifts, migrants may change ascent timing, distance, or participation. These outcomes are plausible and sometimes locally observed, but not uniform predictions for every ocean. Future evidence requires sustained measurements spanning full seasons instead of isolated nights. Autonomous acoustic platforms can track layers without tying observations to occasional ship passages. Targeted net samples must identify organisms while respecting the selectivity of mesh and towing speed. Low-light imaging can document individuals inside layers, though instrument disturbance must remain quantified. Biochemical measurements can estimate feeding and respiration at depth, connecting movement with carbon transfer. Researchers need matched observations of light, oxygen, temperature, predators, prey, and individual condition. The strongest studies will test why some animals migrate, while neighbors remain at depth. They should also resolve larvae, juveniles, and adults rather than averaging incompatible strategies. Satellite observations can map surface conditions, but they cannot directly count animals hidden hundreds of meters below. Models may fill gaps only when their predictions are checked against independent field data. Global biomass totals will remain ranges until broad regions and weak scatterers receive better sampling. The title may endure, but scientific honesty keeps its measurement uncertainty attached. Return now to sunset as the scattering layer loosens from its daytime depth. Fading light lowers visual danger, hungry animals rise, and predators follow the ascending pulse. At the surface, feeding transfers newly produced carbon into bodies destined to descend before morning. When you watch dawn brighten, that living continent is already carrying its night's harvest downward. Its boundaries will shift tomorrow with moonlight, hunger, oxygen, currents, and the arrival of hunters. Diel vertical migration is unquestionably immense, while its exact global size and changing consequences remain unfinished science.